Ah, I think it's working. Maybe. I never quite know. Um, Gosh, how do I get to it? Okay, cool, that is working. And what's the... Okay, pretty cool. So it's nice and sharp. So, right, let me just spread my URL around a bit. Okay, cool. Um, yeah, I can't quite remember everything I've done last week, so um, I think I'm just gonna try and step back through some of what I did, and I've managed to keep all the tabs open too, thank goodness. So I might be able to pick up from where I left off. Actually, you know what? I need to also call this something else. Edit this. Episode 2. All right. Hopefully that's a bit more intelligible. Okay. Yeah, so I was looking at CRISPR. I want to know a bit more about it. So how do you break up a DNA Oh, can you even see this again? So many things to think about. Okie dokie. Yeah. How do you break up a DNA sequence? That was to use CRISPR. How do you put the DNA sequence back together? Well, CRISPR does that on its own. That is the whole mechanic of that. Um, I don't know the symbols yet of chemistry, so need to kind of start learning those at some point. Find a good source of white papers regarding engineering of DNA sequencing. We had a little look. More to come on that. I need to actually set aside some dedicated time for those. I haven't found the inspiration yet. What kind of machines are there? Well, we found that Optitron or Octatron or something like that. What was it called? I'm sure it's here somewhere. Here we go. Oh, Opentron machine. Yep. So let me make sure I've marked that down somewhere as well. And then that was good because it had like a Python API available to it, so I can like move the pipetta from left to right. All these individual um, positions on the board I can get to. Um, I didn't even try to answer that. So is there simulation tooling? And then is there a good database of function over form for like some of the complex words and stuff? Excuse me, that I need to come to learn. So okay, that's cool. So what we found out, you can use adeno-associated virus (AAV) to deliver Cas9 and gRNA to muscle, the nervous system, and other tissues. That was part of that CRISPR stuff. Oh, we've got it here. Look, provides API-driven pipetting machinery. We found this awesome video. Yep, okay, I remember this now. Inject plasmid, apply electrocurrent, electrotransplantation, whatever it was called. Allow the growth. Fact sorting. Yeah, I can't remember what that meant, but I'm sure it's in the keywords. No, it's not. And then ViaCyte was this company who was doing some kind of diabetic therapeutics through using CRISPR. Um, some interesting words, DNA sequencing, SMRT, I can't remember what that is. Yeah, this is that technology, like a third generation technology for DNA sequencing. Okay. Um, the well plate, yeah. Tip rat, yeah. Genetics hat map. I can't remember what that is either. Okay, this is starting to all ring a bell. Um, yeah, contacts. We had that guy from CRISPR itself, so for gene therapy rather than full manipulation of everything between the two join points. Gosh, I can't remember what that meant. Does CRISPR work for gene therapy rather than full manipulation of everything between the two join points? Oh, yes. Yes, okay. So when you remove something, can you then I can't remember what I meant. It's always good to know your notes suck. What size of plasmid injection is needed for DNA sequencing? I think what I meant was like what are the quantities I need to consider. This will decide the size of pipetting tool that I will need to purchase. Yeah, okay. Right. I had so I don't think I'm gonna carry on any of that in specific detail, but what I was kind of getting inspired by was what what are the more detailed processes of using CRISPR? So what chemicals are we going to be dealing with? So let's say this is episode 1, and I'm going to just try and drop all this stuff in there. Oh, if you can. I swear you can. Oh, you can't. Oh, cool, you can. Excellent. Episode 2. Right. Yes, I want to just take a look at the specific materials required for CRISPR. Um, and also DNA sequencing. So materials required for CRISPR and DNA sequencing. And there was one more thing I wanted to do. But so this first one is I really want to start seeing some of the kind of chemical bonds and trying to understand at least some of that. Um, yeah, and I also wanted to just have a little think about um, what I'm really aiming for. Yeah, what am I aiming for as a I don't know product or solution or open source whatever project? Cool. I think that's enough because I'm really tired. But I just thought I had to come on and give it a try. So once again, I'm sorry if you don't ever catch any of these details because I'm not zoomed in. I'm an idiot. So where do we start? Take a look at the specific materials required for CRISPR. I tell you what, one more thing. I'm just gonna just quickly snoop through all this stuff again. Yeah, we had the PowerPoint on CRISPR stuff. Yep. I think we'll need that. Homology is similarity due to shared ancestry. Track. This is that cool database. Kinase was enzyme that catalyzes the transfer of phosphate groups from high energy to specific substrates. Okay. Kinase, high energy to specific substrate. That was that cool dude. Yeah, there he is. Kanamycin, simply as is an antibiotic used to treat severe bacterial infections. FACS sorting, fluorescent activated cell sorting. Yeah, that's one of those. It might have been one of the older generation DNA sequencing tools. So, okay, that's gone into the bank. What does PCR stand for? Yeah, okay. Polymerase chain reaction. Dominant ways that global healthcare systems are testing citizens for COVID-19. That's it, PCR test, right? Confluent, flowing together, great. Um, okay, okay. Single molecule real-time sequencing, SMRT, single molecule real-time sequencing. Parallelized single molecule DNA sequencing method. Single molecule real-time sequencing utilizes a zero-mode waveguide. A single DNA polymerase enzyme is affixed at the bottom of a ZMW with a single molecule of DNA as a template. Okay, SMRT. Actually, probably want to take note of that one. We've got it here already. Cool. That was that cool company. Probably going to use those, to be honest. This was me investigating the pipetting. That's some personal stuff probably. No, maybe. Oh, okay. It was the price for the Opentron thing. Plasmid isolation. Okay. Ah, now this looks good. And I don't remember actually reading through this. And then CRISPR-Cas9 genome editing. Um, these are the papers that I was looking at. Yep, yep. That was Manolis Kellis's stuff. Don't think it's going to be quite what I'm looking for today. Mutagenesis is a process by which the genetic information of an organism is changed by the production of a mutation. It may occur spontaneously in nature or as a result of exposure to mutagens. Okay, that was more of that stuff. And then imputation in genetics refers to the statistical inference of unobserved genotypes. It is achieved by using known haplotypes in a population. Okay, cool. Right, right. So, what are we going to do today then? We just open the window. It's so damn hot in here. Um, let's see. Beginner guide to CRISPR. Let's see what they got. Fill out a short form. We'll send you the comprehensive guide for beginners. Ooh, interesting. Is that going to come up again? No. An ebook. Beginner's Guide to the Powerful Technology that is revolutionizing how scientists edit genomes. That's fair enough. Okay. I don't mind paying for something if we need it. Everything you need to know about CRISPR. That sounds good. Ah, oh, look at this. How lovely. Right. Let's start here. CRISPR-Cas9 has ignited a revolution. CRISPR has enabled a simple and affordable way to manipulate and edit DNA, completely changing the face of genome engineering. The blah blah blah. Okay. Now that CRISPR has gained popularity with the scientific community, there is a surplus of information surrounding this technology. In fact, it can be daunting to scour the internet to identify the useful resources to learn about CRISPR-Cas9. We have compiled the most valuable information to serve as a one-stop resource for all your CRISPR needs, whether you want to brush up the basics of CRISPR. Cool, that sounds awesome. I'll give them a tweet later on as well, not that anyone's following me. CRISPR is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. Clustered Regularly Interspaced Short Palindromic Repeats. Palindromic. Yikes. A nucleic acid sequence in a double-stranded DNA or RNA molecule, wherein reading in a certain direction, e.g. 5, what is that? 5 prime to 3 prime, I don't know what that is, on one strand matches the oh no. Let's just go through to it. Okay. Nucleic acid sequence in a double-stranded DNA or an RNA molecule, wherein reading in a certain direction, e.g. 5 prime to 3 prime, on one strand matches the sequence reading in the opposite direction, e.g. 5 to 3. Okay, that makes sense. So, clustered regularly interspaced short palindromic repeats. CRISPR genome engineering technology enables scientists to easily and precisely edit the DNA of any genome. That's precisely what I want. I'm looking at the right thing. That's a good start. In nature, the CRISPR palindromic repeats play an important role in microbial immunity. When a virus infects a microbial cell, the microbe employs a special CRISPR-associated nuclease, Cas9, to chop off a piece of the viral DNA. The nuclease is directed to its target sequence by a short RNA fragment known as a guide RNA, which is complementary to the target segment of the viral genome. The SNP DNA fragment may then be stored between the palindromic CRISPR sequences to retain a genetic memory for disabling future infections from the same viral strain. The SNP DNA fragment may then be stored between the palindromic CRISPR sequences, to retain a genetic memory for disabling future infections from the same viral strain. Yeah. Okay. Okay. Cool. Once scientists learn how the CRISPR system worked in bacteria, they figured out how to reprogram it to allow efficient editing in any species. Cool. This revolutionary breakthrough has completely changed the way science. Blah blah blah. Want to learn more details? Check out the following resources to get a complete grasp of the CRISPR-Cas technology. This article in Wired answers the "What is CRISPR?" question with an in-depth narration of history, current state, and future applications. Video, ebook. Oh, free as well. That's cool. The comprehensive ebook is the perfect resource for both scientists and non-scientists to learn basics about. Okay, cool. I'm quite interested in that as well. Sites these days are terrible. Come on, that is so ridiculous. I'm all for adverts. Like, I don't mind these, but ones that come up in front of all the content is so annoying. Okay. Future of CRISPR. Uses for CRISPR. History of CRISPR. Cool. And then you got this. Oh, I see. Wait, that's not the link I clicked, is it? No, it's not. This one I want. Oh, it is that one. Interesting. Beginner's guide. Download. Just do this off the screen. Ha. Other. Okay. Download ebook. Let's see if I get a virus instead. Select all images of motorcycles. Sure. Ha! Please enter your business email. Great. Oh, I've already got it actually. That's very naughty of them. Okay, let's see if I can open this up and not show everyone my emails. Awesome. Access it here at any time. Oh, wonderful. Okay. Download. Let's get that bad boy onto my computer. Put it onto my desktop. There it is. Alright, success. Moving this back over. Yeah. So I guess we'll have a little read of this then. Put this in here. Your guide to understanding CRISPR. It's going to be some marketing again. Genome editing involves the deletion, insertion, or modification of specific DNA sequences in the genome. For many years, researchers have been trying to develop easy and cost-effective genome editing tools to address problems across a wide spectrum of fields. For instance, gene therapy in humans could progress rapidly if one could simply eliminate the gene responsible for a certain genetic disorder. Yikes. We know where that goes. In agriculture, manipulating plant DNA could be used to optimize crop yields and control plant diseases. Similarly, bacterial genomes could be fine-tuned to increase their productive yields in several industrial applications. Yeah, this is certainly key to what I'm trying to aim for, but I wonder how I'm gonna. I should probably I should probably consider the ethics, or at least the questions that are raised about this. I mean, we all know about what Hitler got up to with epigenetics. I am reading The Rise and Fall of the Third Reich because Lex Fridman had been reading it. I was very intrigued to kind of all the history that I wouldn't have learned at school. So it's good to know your enemy, I guess. But I think from a very high end perspective, whatever it's called, um, removing things that cause pain and suffering sounds like a positive thing, but there may be reasons why it's not a good idea to do that kind of thing. So definitely worth thinking about that. There's a lot to think about actually. But I suppose from a technical point of view, that's kind of once I start seeing a horizon come up, then I will need to start ensuring that I work responsibly. Okay, yeah. Finally, the efforts of researchers paid off with the development of CRISPR, a robust molecular tool that can edit DNA at virtually any locus. CRISPR technology is igniting a revolution across the life sciences and is quickly becoming a standard tool in many labs. Given its ease of use and versatility, CRISPR is already being used for a variety of applications and holds a lot of promise for the future. Read on for a crash course. When was this written? 2019. Cool. Okay, that's promising. Genome editing tools before CRISPR. Although CRISPR has now become synonymous with gene editing, it is not the first technology developed to edit DNA. Genome editing techniques initially emerged with the discovery of restriction enzymes and meganucleases. Nucleases. The possibility of precision gene editing was made clearer with the discovery of zinc finger nucleases. Zinc finger. That's interesting. The possibility of precision gene was made clearer with the discovery of zinc finger nucleases. I think I've seen this or heard of this before. Not ink finger. Small protein structural motif that is characterized by the coordination of one or more zinc ions. Zened squared. Uh, Zened to the power of 2. In order to stabilize the fold. So Zened squared. What was that trying to say? I'm interested in one more zinc ions. Zen N2. I don't think that's quite what I think it means. N2. I wonder if this is the. I really want to know what I'm looking at there. Two charge. Ah, here we go. It might be this, you see. I don't think it's a mathematical term. Yeah, two positive charges in the nucleus. Haha, okay, that might be right. Oh, it's hard to tell straight away. I might have to come back to that. Might have to come back to that. Okay, cool. In order to stabilize the fold, it was originally coined to describe the finger-like appearance of a hypothesized structure from the African clawed frog. Ah, that's interesting. Wow, that is so intriguing. Transcription factor 3A is a protein that in humans is encoded by the GTF3A gene. It was first isolated and characterized by Wolf and Brown, 88. It has been found to encompass a wide variety of differing protein structures in eukaryotic cells. These are all things I've heard of. It's really weird. I don't know how. I just don't know what they are. Eukaryotic? Yeah, I don't know how you spell this actually. Say this. Are organisms whose cells have a nucleus enclosed with a nuclear envelope. A eukaryotic cell. Eukaryotes are organisms whose cells have a nucleus enclosed with within a nuclear envelope. This is a great diagram here. I don't know if you can see this. Roughly. Nuclear envelope, outer membrane. Yeah, okay. I know this stuff. That's all good. Nuclear pore, ribosomes. Cool. Okay. That's fine. The ZFN, the zinc finger nucleases method, involves engineering an enzyme with both a zinc finger DNA binding domain and a restriction endonuclease domain. The zinc finger domain is composed of three base pair site on DNA designed to target and bind to specific sequences of DNA, and the nuclease domain cleaves the DNA at the desired site. DNA designs and the nuclease domain cleaves the DNA at the desired site. Okay. Although zinc finger editing represented the first breakthrough in site-specific genome engineering, they have several limitations. Okay. In addition to exhibiting off-target effects, ZFNs are expensive and time-consuming to engineer. Furthermore, their inefficiency limits their practical application to only one genomic edit at a time. Many years after zinc fingers made their debut, a similar method known as transcription activator-like effector nucleases, TALENs, was developed. This I've heard on a podcast. The TALENs method utilizes engineered enzymes containing a DNA binding domain and a separate DNA cleaving domain. Okay, well, so the TALENs utilizes engineered enzymes containing a DNA binding domain. Okay, and a separate DNA cleaving domain, similar to the ZFN's method. However, TALENs have an advantage over ZFNs because they are more flexible. The DNA binding domains can target a wider range of sequences. Although they are easier to design than the ZFNs, TALENs are expensive to produce. Okay. An additional genome editing technique uses engineered restriction enzymes. Enzymes in concert with recombinant adeno-associated viruses, RAVs. AAV is a non-pathogenic virus that infects mammalian cells at all stages of the cell cycle and integrates into the host genome at predictable sites. The adeno-associated virus genome can be modified to target specific sequences in the host genome and integrate desired DNA sequences. Okay, that's great. That is really, really cool. I think we were a bit excited about it last time. to use the viruses to get in. I remember having a very, very, very super brief conversation with my brother's wife about this. I remember it. However, the AAV approach also has several limitations. For instance, the vectors are difficult to produce and can only accommodate a small amount of genetic material. Up until now, the field of genome engineering has provided researchers with a few gene editing technologies, all of which have limitations. Table 1. Okay, so let's have a look. CRISPR, low cost, complexity easy, multiple edits easy. What was that again? Antivirus, oh no, virus something. Where is it? AV. Adeno associated viruses. Talon's was transcription, activator like, effector nucleases. Sheesh. Okay. Zinc fingers. Because ZFNs and talons require complex protein DNA interactions, they are challenging to design and manipulate. Oh my god, are you kidding me? I've forgotten already. God, the brain's so dumb. My brain's so dumb. Literally just read it. It's so annoying. Adeno associated. Adeno associated viruses, vectors, are also difficult to work with and have limited applications given their small packaging capacity. CRISPR, which relies on well understood interactions between DNA and RNA, offers a far simpler way of editing genes and has completely changed the face of genomic engineering. Okay. Adeno-activated virus. CRISPR is a simple two-component system that can be used to target a specific genomic sequence and then make a cut in the DNA. This powerful tool enables scientists to manipulate a specific gene of interest. For instance, one may knock out a gene, making it inactive, or knock in a particular sequence that repairs a mutation or adds a characteristic. Below, we describe how CRISPR was discovered and developed into a powerful genome editing tool. Okay. The foundational discoveries that led to the development of CRISPR, Cas9 technology, can be traced back to 93, when repetitive palindromic segments of DNA interspaced with other fragments of genetic material were identified in prokaryotes, whatever it's called. How do you say this? How do you say? Come on, let's see if this lets us down again. Prokaryotes. Prokaryot. I don't know. We'll find out. No, we will not. How do you say? There's been two words like this now, so I'm quite interested to know how this sounds.
Prokaryotes.
Prokaryotes. Prokaryotes. Interesting. Glad I looked that up actually. Prokaryotes. These repeated segments of genetic code were named clustered regularly interspaced short palindromic repeats, or CRISPR. However, the segments of DNA between the repeats were really the interesting part. These repeated segment of genetic code, CRISPR, however, the segments of DNA between the repeats were really the interesting part. In 2007, after years of study, researchers concluded that CRISPR's function is related to prokaryotic immunity. Prokaryotic. Prokaryotic. Prokaryotes. Wait, what was that again? Undo close tab.
Prokaryotes.
Prokaryotes. Okay, we'll get that. Prokaryotic. It took the combined efforts of several research groups over the next 5 years to elucidate the underlying molecular mechanism behind the CRISPR system. Okay. As it turns out, bacteria and archaea, oh god.
Archaea.
Archaea, okay. As it turns out, bacteria and archaea use the CRISPR-Cas9 system to defend themselves against invading viruses. Bacteria and archaea use the CRISPR-Cas9 system to defend themselves against invading viruses. called bacteriophages. Upon encountering a viral infection, the prokaryotic cell employs a special CRISPR-associated nuclease, Cas9, to snip off a piece of viral DNA by creating a double-strand break in its target loci. How does the Cas9 protein recognize the target DNA? It is directed to the target sequence by a short RNA fragment known as a guide RNA. The guide RNA is complementary to a segment of the viral genome, which allows Cas9 to cleave DNA with a high degree of specificity. Not only does cleaving the DNA destroy the virus, but a fragment of foreign DNA called a spacer may be stored between the palindromic sequences of the CRISPR array as a way of retaining a genetic memory of past infections. If the virus were to reinvade, the CRISPR-Cas9 system could quickly target and destroy it. This library of viral fragments is thus essentially equivalent to our immune system, okay, which stores antigens to prepare for future infections. That's awesome. I'm just going to double check YouTube here. I'm really interested in recording this for myself, essentially. Let's see. Okay, so it does come up as DNA. That's great. Good stuff. Have a quick break. Let me see what's going on with my phone. Work stuff. Okay, cool. That's really interesting. Once scientists figure out the mechanism for CRISPR in prokaryotes, it did not take long for them to realize the potential for engineering the genomes of microbes, plants, and animals. Today, CRISPR is utilized for a variety of applications as adoption continues to increase in laboratories throughout the world. So 2007, what was it again? Yes. After years of study, research concluded that CRISPR's function is related to prokaryotic immunity. So they started to understand it then. Can we trace back to 93? Okay. Cool. Okay. The components of CRISPR. The CRISPR system comprises of two components: a guide RNA that is specific to the target DNA sequence. This is something I'm going to need to engineer. That is what I'm going to write down. New page. Let's just call it thoughts for now. It doesn't really matter. I just find that writing it down, even though I may never come back to it, just kind of helps me to embed it in my head a bit. So gRNA will need to be engineered for the usages I require. Okay. For CRISPR experiments, the guide RNA and Cas endonuclease are combined to form a ribonucleoprotein (RNP) complex. Complex. I wonder what that means in it's like Latin or something, is it? What am I trying to do here? Etymology of complex. Complexure. Embrace. Complexus. Plated. Complexus. Complex. Group of related elements. Okay, that makes sense. The Cas protein functions as a pair of molecular scissors, while the gRNA is the GPS that guides it to the appropriate site. Ha! In prokaryotes, the gRNA guides the nuclease to viral DNA, but as a bio... Oh, whoa. Sorry, I'm going to have to say this again. So the Cas protein functions as a pair of molecular scissors, while the gRNA is the guide to the appropriate site. In prokaryotes, the gRNA guides the nuclease to the viral DNA, but as a biotechnological tool, the design specifications of the gRNA can be altered to target any organism's genome at virtually any location. This is it. This is exactly what I want to hear. Keep telling me things I want to hear. For many genome engineering applications, the Cas9 protein from Streptococcus pyogenes is used. For many genome engineer applications, Streptococcus. Let's have a look at this then. Pyogenes? I don't know. Pyogenes? How do you how do you say
Streptococcus pyogenes.
pyogenes? Okay,
Strep.
cool. Oops. Streptococcus pyogenes is a species of gram-positive aerotolerant bacterium in the genus Streptococcus. These bacteria are extracellular. These bacteria are extracellular, outside of the cell. Let's just God, I hate Latin. I like Latin. I'm joking. Extraordinary. Uh, what does extra mean in biology? Beyond outside. I thought so. These bacteria are outside cellular and made up of non-motile and non-sporing cocci. It is cocci. That's probably wrong. It is clinically important for humans. It is an infrequent but usually pathogenic part of the skin microbiota. Microbiota. I don't know. It is the predominant species harboring the Lancefield group A antigen. So this seems really important if it's, like it said, the main, one of the main. Oh no, for many genome engineering applications, this dude is used. Okay. Thoughts and notes. Let's do another page. Make sub page. Ah! Notes. Streptococcus. Yeah, nice. Pyogenes is pretty important for Cas9, used often for CRISPR. Binding of the gRNA to the genomic target is contingent upon the presence of a short protospacer adjacent motif PAM. Binding of the gRNA to the genomic target is contingent upon the presence of a short protospacer adjacent motif located directly downstream of the target on the opposite DNA strand. Okay. So let me try and understand this. Binding of the gRNA to the genomic target, so the thing that we tell the gRNA to go and target, is contingent upon the presence of this thing, a protospacer adjacent protospacer adjacent motif located directly downstream of the target, i.e., or meaning on the opposite DNA strand. I have this funny. So I don't quite know, but I think the DNA strands are like duplicates. They're symmetrical. So this is interesting. This is interesting. I think so. DNA strands symmetrical. Yeah, not so sure, but good to think about. The Cas proteins from different prokaryotic species recognize different PAM motifs. The PAM for Cas9 is 5. Okay, okay. Let's get into this. This is the stuff we want. This is the stuff I guess you learn at uni. Ooh, nice. Well, that's not what I wanted, but we'll go back to it later. PAM in CRISPR, the protospacer adjacent motif, or PAM. Oh, is, is this helpful? I think I prefer wiki for this one. Is a 2 to 6 base paired DNA. Now, why would it have the hyphen if it wasn't 2? So maybe it is 2 to 6, but let's just say it's 2 6 base pair DNA sequence. So protospace adjacent motif is a 2-6 base pair DNA sequence immediately following the DNA sequence targeted by the Cas9 nuclease in the CRISPR bacterial adaptive immune system. Let me read that again. A protospace adjacent motif is a 2-6 base pair immediately following the DNA sequence targeted by the Cas9 nuclease in the CRISPR bacterial adaptive immune system. The PAM is a component of the invading virus or plasmid, but is not found in the bacterial host genome and hence is not a component of the bacterial CRISPR locus. Cas9 will not successfully bind to or cleave the target DNA sequence if it is not allowed by the PAM sequence. Oh! PAM is an essential targeting component which distinguishes bacterial self from non-self DNA. PAM is an essential targeting component which distinguishes bacterial self from non-self DNA, thereby preventing the CRISPR locus from being targeted and destroyed by the CRISPR-associated nuclease. Ah, that's really good to know. PAM is good to know. Let's get more of PAM. PAM seems pretty important to understand. Awesome. Okay. The PAM for Cas9 is 5-NGG-3, where N is any nucleotide. If a correct PAM match is made, and if the gRNA successfully binds to the target, then Cas9 cleaves both DNA strands, 3 to 4 nucleotides upstream. Okay, whoa. If the correct PAM match is made, and if the gRNA successfully binds to the target, then Cas9 cleaves both DNA strands, both DNA strands nucleotides upstream of the PAM site. In nature, this short genetic element only occurs in invading viruses, not the bacterial genome. Oh, okay. And thus ensures that Cas9 does not cleave its own CRISPR locus. Let's check this diagram out. Figure 1. The CRISPR-Cas9 system. The CRISPR-Cas9 system comprises a guide RNA and Cas9 nuclease, which together form a ribonucleoprotein RNP complex. Okay, let's see. CRISPR-Cas9 guide RNA. Cas9 nuclease. Target DNA. Oh. Oh, I see. This diagram's going to make more sense later on. Yes. Okay. This is really good. I'm really glad I'm doing this. This is really helpful. The presence of a specific protospace adjacent motif in the genomic DNA is required for the gRNA to bind to the target sequence. The Cas9 nuclease then makes a double strand break in the DNA. The Cas9 nuclease then makes a double strand break in the DNA, denoted by the scissors. Okay, I think I need to see one of these cells as well. These nucleases in reality. I need some kind of concrete example. Endogenous, endogenous, I can't remember what that. Indigenous, endogenous, I think it's probably that. How do you say?
Endogenous.
Endogenous. Okay, cool. I didn't know that. Endogenous repair mechanisms triggered by the double-strand break may result in gene knockout via a frame shift mutation or knock-in of a desired sequence if a DNA template is present. Okay. Endogenous repair mechanisms triggered by the double-strand break. Okay. Endogenous repair mechanisms triggered. So, oh, I see. So when it snips it, it may result in gene knockout via a frame shift mutation or knock-in of a desired sequence if a DNA template is present. So we talked about this on the first thing. This is where you cut it out and use the body's own tools to try and look for that same part that was taken out. So there's more to this. There's more in this paragraph, but I'm going to get back to it. Guide RNA, the RNA component of the CRISPR-Cas9 genome engineering tool, contains a roughly 20 base pair sequence that is complementary to the genomic target. Roughly 20 base pairs. Okay. This doesn't sound super helpful. Base pair sequence. It's very specific. Okay. I guess we'll learn about that more. CRISPR CRISPR-associated endonuclease protein, Cas, a family of proteins that is able to cleave nucleic acids. Cas9 is the most commonly used Cas protein in CRISPR experiments. So there's different ones. Protospacer adjacent motif, a short sequence of nucleotides that must be present downstream of the target site in order for the nuclease to make a double-strand break. I wonder if this causes problems because you're looking for specific things, or is it that you're putting those things in? Are you engineering these, or are they required for this process to work? Okay. What's the time? 8:01. When did I start? How long have we been on? Does it say? 1 hour nearly. Okay. I don't want to go for too long. Okay. Guide RNA formats. In its natural form, the gRNA consists of two distinct segments of RNA, CRISPR RNA and transactivating CRISPR RNA, a tracer RNA. Cool. The cRNA is an 18 to 20 base pair sequence that binds the genomic target. Okay, fine. So I think we need to know what this base pair means properly. Base pairs. A base pair is a fundamental unit of double-stranded nucleic acids consisting of two nucleobases bound to each other by hydrogen bonds. They form the building blocks of the DNA double helix and contribute to the folded structure of both DNA and RNA. Cool. Okay, let me read that again. A base pair is a fundamental unit of double-stranded nucleic acids, okay, consisting of two nucleobases bound to each other by hydrogen bonds. Cool. They form the building blocks of the DNA double helix and contribute to the folded structure of both DNA and RNA. Dictated by specific hydrogen bonding patterns, a Watson-Crick base pairs, guanine, cytosine, and adenine thymine, allow the DNA helix to maintain a regular helical, I don't know what that is. Cool. Okay, this. structure that is subtly different on its nucleotide sequence. Let me read this again, my apologies. Dictated by specific hydrogen bonding patterns, Watson-Crick base pairs allow the DNA helix to maintain a regular helical structure that is subtly dependent on its nucleotide sequence. The complementary nature of this base pair structure provides a redundant copy of the genetic information encoded within each strand of DNA. The regular structure and data redundancy provided by the DNA double helix make DNA well suited to the storage of genetic information, while base pairing between DNA and incoming nucleotides provides the mechanism through which DNA polymerase replicates DNA and RNA polymerase transcribes DNA into RNA. Many DNA binding proteins can recognize specific base pairing patterns that identify particular regulatory regions of genes. There is a lot in that. I didn't quite catch it all. Let me just mute myself whilst I drink. Okay. Okay. Yeah, let's try this again. It's a bit heavy. The complementary nature of this based pair structure provides a redundant copy of the genetic information encoded within each strand of DNA. That makes complete sense to me. The regular structure and data redundancy provided by the DNA double helix make DNA well suited to the storage of genetic information. I guess because if one gets knocked out, the other one's still there. While base pairing between DNA and incoming nucleotides provides a mechanism through which DNA polymerase replicates DNA and RNA polymerase transcribes DNA into RNA. Transcribes DNA into RNA. Okay. So while base pairing between DNA and incoming... Okay, fuck. I can do this. Come on. This makes some sense. The complementary nature of this based paired structure provides a redundant copy of the genetic information encoded within each strand of DNA. Yes. The regular structure and data redundancy provided by the DNA helix make DNA well suited to the storage of genetic information, while base pairing between DNA and incoming nucleotides provides the mechanism through which DNA polymerase replicates DNA. So this allows the replication of DNA, and RNA transcribes DNA into RNA. Okay, that makes sense. Many DNA binding proteins can recognize specific base pairing patterns that identify particular regulatory regions of genes. Wow, this is interesting. In molecular biology, RNA polymerase, RNAP, is an enzyme that synthesizes RNA from a DNA template. Okay, using the enzyme let's have a look at this one.
Helicases or
Helicases. Heli or heli. Okay, helicases. Okay, cool, that's fine. So using the enzyme helicase, RNAP locally opens the double-stranded DNA so that one strand of the exposed nucleotides can be used as a template for the synthesis of RNA, a process called transcription. Okay, a transcription factor and its associated transcription mediator complex must be attached to a DNA binding site called a promoter region before RNAP can initiate the DNA unwinding at that position. This is the stuff. Using the enzyme helicase, RNAP locally opens the double-stranded DNA so that one strand of the exposed nucleotides can be used as a template for the synthesis of RNA. One strand. Oh, okay, I see. So, I see. So when it gets cut into the parts. No, so when the DNA is cut, one strand of it using helicase exposes the nucleotides so that it can, so one of them could be used as a template for the synthesis of RNA. Okay, I see. Ooh, that's cool. So let me just try and draw that, get it in my head. That's not what I want. So I'm imagining something like this. So you've got your double helix. Right. And when it cuts it, when it cuts it, uh, process called transcription. So using the enzyme helicase. So helicase RNA polymerase locally opens the double-stranded DNA. Okay. Using the enzyme RNAP. It's an enzyme that synthesizes using the enzyme helicase. Okay. So this is the RNA polymerase. Now I want to know what that word really means. So what's the etymology of that? Enzyme, yeah, that's not helpful. Enzyme which catalyzes the formation of a polymer, polymer. Okay. Enzyme which catalyzes the formation of a polymer. So is it the same word? Can't be. Damn. Okay, fine. An enzyme which catalyzes the formation of a polymer. What's a polymer? Many. Haha, got ye. Merase, a share. Many, shared. Having many parts. Haha, yes. Okay, cool. I like it. So having many parts is an enzyme that synthesizes RNA and DNA template. Cool. Using the enzyme helicase, RNA polynase locally opens the double-stranded DNA. So let me go back to my image. So this is how it starts. And then we've got, oh, is anyone communicating to me? I completely forget to check these things. No, okay, cool. Yeah, cool. Okay, it's just me. This now opens up. Okay, so let's imagine this goes on a bit longer, like that, and we are cutting here. Then this part becomes like this, and that becomes like that. Using helicase, RNAP locally opens the double-stranded DNA so that one strand of the exposed nucleotides, this one, can be used as a template for the synthesis of RNA. That's so cool. Okay. Template. That is so cool. Okay. A process called transcription. So this whole thing is transcription. Okie dokie. Right, we're getting somewhere. Transcription. The helicase is the RNA polymerase. It cuts the DNA, it leaves them open. Because it's double-stranded, one of them can be used as a template for the synthesis of RNA. That makes sense. And this whole thing's called transcription. A transcription factor and its associated transcription mediator complex must be attached to a DNA binding site called a promoter region before RNAP can initiate the DNA unwinding at this position. So a transcription factor and its associated transcription mediator complex must be attached to a DNA binding site called a promoter region before RNAP can initiate the DNA unwinding at that position. So this process cannot happen transcription factor and its associated transcription mediator must be attached to a DNA binding site called a promoter region. So something has to be attached. Let's put this in. Let's go. let's go lighter green. Something has to be attached here. And this is the mediator complex, a transcription factor, and its associated transcription mediator complex must be attached to a DNA binding site called a promoter region. So this is the promoter region. Promoter region. And then that must have the mediator complex and the transcription factor applied to it first. So the promoter region requires the transcription factor and the mediator complex. Okay, good. That makes sense. RNAP not only initiates RNA transcription, it also got. Is this an automatic process as well? It also guides the nucleotides into position, facilitates attachment and elongation, has intrinsic proofreading and replacement capabilities, and termination recognition capability. In eukaryotes, RNAP can build chains as long as 2.4 million nucleotides long. Okay, cool. Okay. Let's see what this is. This seems super important. A transcription factor or sequence-specific DNA binding factor is a protein that controls the rate of transcription of genetic information from DNA to messenger RNA by binding to a specific DNA sequence. I'm going to read it again. In molecular biology, a transcription factor is a protein that controls the rate of transcription of genetic information. Protein that controls the rate of transcription of genetic information from DNA to messenger RNA. It controls the rate of it. So there's a bandwidth here. In molecular biology, a transcription factor or sequence-specific DNA binding factor is a protein that controls the rate of transcription of genetic information from DNA to messenger RNA by binding to a specific DNA sequence. Oh god, it's another thing that might come in as a problem. So there is a bandwidth problem here. Not problem. There is a bandwidth. Let's go to this is a bandwidth. I think what I'm going to do is start to model some of these in code. I really want to get a grasp on these specific details. Bandwidth of the transcription of genetic information from DNA to messenger RNA. Bandwidth for DNA to RNA. Yeah, okay. The function of TFs is to regulate turn on and off genes in order to make sure that they're expressed in the right cell at the right time. So they have a very specific function and in the right amount throughout the life of the cell and the organism. What controls this? Sheesh. Is it its own mechanism? Don't you hate it when you've been reading and you just kind of lose the place and you've got to read the whole bloody thing again? I'd love to have some kind of, as you're reading it, it's highlighting the words and then it kind of leaves a very subtle fading that goes away. Then you know what you've already read. Okay, so the function of transcription factors is to regulate or turn on and off genes in order to make sure they're expressed in the right cell at the right time and in the right amount throughout the life of the cell and the organism. Groups of transcription factors function in a coordinated fashion to direct cell division, cell growth. Groups in a coordinated fashion. Jeez. Okay. Okay. So fashion to direct cell division, cell growth, and cell death throughout life. Cell migration and organization body plan during embryonic development and intermittently in response to signals from outside the cell, such as a hormone. There are up to 1,600 transcription factors in the human genome. Transcription factors are members of a proteome as well as a regulome. That word doesn't sound right. How do you say that?
Regulome.
Ah, okay, regulome. Fair enough. Weird. I don't like it. Um. Okay. Okay. So let's try and get back to the track we were on. Base pairs. That's right. So I'm going to read this one more time. I think I'm going to get to the end of my learning session here. So a base pair is a fundamental unit of double-stranded nucleic acids consisting of two nucleobases bound to each other by hydrogen bonds. They form the building blocks of the DNA double helix and contribute to the folded structure of both DNA and RNA. Dictated by specific hydrogen bonding patterns, Watson-Crick base pairs allow the DNA helix to maintain a regular helical structure that is subtly dependent on its nucleotide sequence. So it's okay, so it bends because of the nucleotides in that sequence. That makes a lot of sense. Okay, probably some physics in there actually. The complementary nature of this base paired structure provides a redundant copy of the genetic information encoded within each strand of DNA. The regular structure and data redundancy provided by the DNA double helix make DNA well suited to the storage of genetic information, while base pairing between DNA and incoming nucleotides provides the mechanism through which DNA polymerase replicates DNA and RNA polymerase describes DNA into RNA. That makes complete sense now. Many DNA binding proteins can recognize specific base pairing patterns that identify particular regulatory regions of genes. Okay, cool. Right, I'm a bit hungry. 10 more minutes. Intramolecular base pairs can occur within single-stranded nucleic acids. Intramolecular base pairs can occur within single-stranded nucleic acids. This is particularly important in RNA molecules, e.g. transfer RNA, where Watson-Crick base pairs, guanine, cytosine, adenine uracil, permit the formation of short double-stranded whatever it is, and a wide variety of non-Watson-Crick interactions, e.g. GNU or ANA, allow RNAs to fold into a vast range of specific three-dimensional structures. In addition, base pairing between transfer RNA and messenger RNA forms the basis of the molecular recognition events that result in the nucleotide sequence of mRNA becoming translated into the amino acid sequence of proteins via the genetic code. What? Okay, let me just read that again. Intramolecular base pairs can occur within single-stranded nucleic acids. Okay, this is particularly important in RNA molecules, where Crick base pairs permit the formation of short double-stranded whatever it is. I need to know this because it's fucking me up. I've already bloody looked it up. How do you say
Helices.
helices? So annoying. Okay. Intramolecular base pairs can occur within single-stranded nucleic acids. This is particularly important in RNA molecules, where Watson-Crick base pairs permit the formation of short double-stranded helices, and a wide variety of non-Crick interactions allow RNAs to fold into a vast range of specific three-dimensional structures. Okay. In addition, base pairing between transfer RNA and messenger RNA forms the basis for the molecular recognition events. In addition, base pairing between tRNA and mRNA, forms the basis for the molecular recognition events that result in the nucleotide sequence of mRNA becoming translated into the amino acid sequence of proteins via the genetic code. Okay, that's interesting. I'm getting there. I'm starting to feel this a bit. I'm starting to feel you. Okay. The size of an individual gene or an organism's entire genome is often measured in base pairs because DNA is usually double-stranded. Hence, the number of total base pairs is equal to the number of nucleotides in one of the strands. Okay, the size of an individual gene or an organism's entire genome is often measured in base pairs because DNA is usually double-stranded. Hence, the number of total base pairs is equal to the number of nucleotides in one of the strands. Huh, that's interesting. With the exception of non-coding single-stranded regions of telomeres. Oh, that's so interesting. The haploid human genome, 23 chromosomes, is estimated to be about 3.2 billion bases long and to contain 20k to 25k distinct protein coding genes. A kilobase is a unit of measurement in molecular biology equal to 1,000 base pairs of DNA or RNA. The total number of DNA base pairs on Earth is estimated at 5 times 10 to the 37 with a weight of 50 billion tons. Yikes. In comparison, the total mass of the biosphere has been estimated to be as much as 4 trillion tons of carbon. Okay. This, I need to read this again. And I think I'm going to leave this for another day. This has been amazing. I want to get back to this. So, this is where I am. I'm going to prepare my episode 3, I think, because I know what I want to do in the next one. Continue reading the CRISPR 101 ebook and continue investigating the base pairs and the base pairs. Yeah. Okay, I'm going to call it a day. That was awesome. That was really awesome. Hope this all recorded okay. And I will speak to you next time. I guess I'm going to probably do it Friday unless I get a bit tired. So maybe Thursday or Friday. No, definitely not tomorrow. Maybe Saturday, Friday or Saturday. Something like that. Bye for now.